today welcome to Professor David Healy, who has agreed to join us from, I think, Canada today. And we are very, very pleased to have you here today. I don't want to say very much more. I hope you can introduce yourself a little bit. I just want to also acknowledge that André Marx is our contact with you. He has arranged this and reached out to you, and we're anyway very glad to have you here.
Great, thank you very much. Okay, hopefully you can all see the screen. Actually, I just turned me off as well, so let's leave me up there in the upper corner just for a moment. If you want to know more about me, there will be some material at the end of the talk, which will give you some hints about where I've come from and what I do. The key thing you need to know at this point in the talk is that I'm a very orthodox doctor. Okay, I'm very conservative. I believe in the medical model. I believe in physical treatments for mental illness and all illnesses. So it's not going to get a talk from a person who's radical. Okay, so, but I think you are, even though it's a very conservative talk, I think you're probably going to be disturbed, and disturbed possibly by the distance we seem to have come from what used to be called medicine, and disturbed also maybe by where we seem to be going. Now, I'm not going to offer you any answers to where we're going, but I will be asking two questions and offering answers to those. One is, what are RCTs? What are randomized controlled trials? And the other thing I'll be asking is, what is data? Now, you're hopefully going to be able to get the text of the talk and all of the slides afterwards. So there's no great need to pay too much heed in the sense of take notes and things like that. Just listen and appreciate the cartoons. There will be a lot of cartoons and there'll be some humor in the talk also. Right, let me move on to the first slide. We, that is you and me, we have a person who's just come for help from us. They're in the waiting room outside our office. And it's very hard to tell if this person is male or female. It's hard to tell how old they are. They smell a bit, okay? And the other thing about them is it doesn't really look maybe as though they're all that bright. At least that's what the other people in the office who've been waiting to see you come in and say to you, look, there's this person out in the office who doesn't seem all that right to us, okay? So the issue is we're going to have to help this person. And by help, I don't mean tell the person what to do or how to live their life. I mean something else, as you'll see. But if we talk about the kind of help doctors can give, the conversation always immediately turns these days to randomized controlled trials, RCTs. And if anyone talks about RCT, they usually, if they're talking about the history, they mention this man, Ronald Fisher, who's supposedly the person who kicked it all off. He was the person who introduced the concept of randomization. Fisher was a cantankerous that not everybody liked. He was also not a scientist, he was not a doctor, and he didn't ever run an RCT. What he was a person doing maths, and he was keen to try and work out how mathematically we can characterize the views of experts. Fisher thought experts generally were people who knew what they were doing, and there was very little that they didn't know But in terms of an expert running an experiment, they'd run the experiment to demonstrate that they knew what they were doing. They didn't run experiments to find things out. They ran them to show that they knew what they were doing. And in terms of running the experiment, only two things could get in the way. If for some reason there was some confounder that nobody knew anything about, and randomization was supposed to take care of that. And the other thing was chance, as you'll see. Now, for Fisher, an expert was a person a little bit like Robin Hood. They knew what they were doing, so when they took aim at the target, the second arrow would split the first arrow. They would always get exactly the same result. Randomization, as I said, took care of the unknowns the expert might not know about, but these were going to be extremely few. But there could be a gust of wind, something like that came by chance. So Fisher said, well, we can agree that the expert knows what they're doing if they get the same result 19 times out of 20. If they miss once because of a gust of wind, well, we still figure this person knows what they're doing. If any of you can find any control trials of any drug anywhere in medicine for any reason where you get a result like this, please let me know. Because as far as I'm concerned, the control trials for antidepressants and blood sugar lowering agents or any drugs you care to think about that we use look more like this. The results don't repeat themselves. They are all over the target. And there's loads more results other than just these. There are the ones that don't hit the target at all that you don't get to hear about. So something is wrong with RCTs. Fisher's expert was someone more like the person who said to you, if you're going to jump out of a plane, I'd prescribe a parachute, which, is a good idea, but there's very few treatments in medicine that are like parachutes. And if there were, well, we don't need RCTs to let us know what to do. It's just common sense. So Fisher, as I said, was not a man that lots of people liked. And two of the people who least liked him were called Jerzy Neyman and Egon Pearson, whose names you see down here. And they said, Fisher has it wrong. Statistical significance is all wrong. That what we should be doing in experiments is looking at what Carl Friedrich Gauss did. And you see Gauss here on this old Deutschmark note, celebrating his major discovery, or one of his major discoveries, which was the idea of the confidence interval. And you see the confidence interval there just beside his right cheek. And what the confidence interval was about was Gauss was solving a problem that astronomers had around 1800, which was when they looked through the telescopes they had back then, they couldn't be sure if they were seeing one or two stars, that the measurements weren't always quite where they thought they should be. And Gauss solved the problem by saying, well, if the reason the star doesn't seem to be in quite the same place each time, and it's down to the telescope, then the different measurements will be random, and you can map them under a confidence interval. And if you get a measurement that falls outside that, well, then it probably comes from a different star. And Jerzy Neyman said, this is what we should be doing in experiments. Now, this was before anyone had ever done any RCTs in medicine. 50 years ago or so, most medical journals said, well, actually, Neyman and Pearson are right. We shouldn't be doing what Fisher said. We should be using confidence intervals instead of statistical significance. But there's a problem here, and let me show you. Gauss's confidence intervals are up there on the top. And let's say you gave a beta blocker to a person. Let's say I gave Andre a beta blocker. His heart rate would slow down. And we can map the different measurements of his pulse, which wouldn't always be quite the same. We could map them under confidence interval, and the confidence interval would be to the left of that black line you see in the middle, which is the 1.0 line. But if we repeated the experiment and gave a beta blocker to 10 people, or maybe 100 people, there would for sure be one person whose heart rate speeds up. And their pulse rate, their heart rate, would fall on the opposite side of the 1.0. It wouldn't be under the same confidence interval. And we should be saying, well, look, there's 2 stars here, but we don't. We don't say there's 2 stars, partly because this is quite rare when a person's heart rate speeds up like this. But there's lots of drugs we use, particularly in the mental health field, that will cause some people to become sleepy. But the same drug will cause a lot of other people to be more awake and unable to get to sleep. And if you map those under confidence interval, what you get is an interval that looks like the interval here in Gauss's curve, with half of the measurements falling on the right-hand side of the 1.0 line and half falling over on the left. And the pharmaceutical company saying, look, this just proves that our drug has no effect on sleep at all, which is crazy. It's just not right. It shows that confidence intervals don't work for a lot of the things that the drugs we use do. Let me give you a different example. Down on the lower left, and I have to say to you, this is the most complex slide we have. After this, they'll get much easier. But on the left, the lower left, you've got the results from 2 different drugs, both of which can kill you. There's the red drug and the yellow drug. Now, if you're asked which of these drugs, I mean, if you're told you have to take one of these drugs and you're asked, which do you want to take? All of the books and all of our training would say to you, you should take the red drug because it's the one that we know for sure can kill you. The yellow drug, we don't know for sure. The problem is, if it does kill you, it's much more likely to kill you. And the answer is, if you're forced to take one of these drugs, you should take the yellow drug. That what the books tell us about what these data mean is just wrong. And let me bring that out by showing you the graph on the right. Again, you've got a yellow drug and a red drug here, but actually they're both the same drug. The difference here is the ages of the people they've been given to. The red curve there is antidepressants being given to teenagers and the suicidal events that happen to these teenagers. And we're told that, yes, there is evidence that antidepressants can cause teenagers to become suicidal, but there isn't any evidence that anyone else becomes suicidal. And that's the yellow drug. This is people in the age range 45 to 65, where you see that the yellow line goes over the 1.0 line. And this means, according to a lot of people, that there's no real evidence that these drugs cause people to become suicidal, when in actual fact, the common sense interpretation of this is that it's just as likely to cause people to become suicidal as it does in teenagers. So let me move on. My point ultimately here is this, that you can do randomized controlled trials, and we've got tens of thousands of them now in the various different drugs we use, but we have no way to reliably interpret the results that we get from them. Everything you're told in the books, it says, do this and do that, and you'll get a cast iron answer. This is something you can depend on. The books are wrong. So you're all going to get all of the slides. And one of the things you can do when you get the slides is you can print off this slide and you can cut out the tabletops and superimpose one on the other, and you'll find that they're exactly the same size and shape. Now, this doesn't look to be the case to you and me, but it is. And we're going to walk through some odd randomized controlled trial results, which will leave you as confused as the tables here leave you. Here's the first randomized controlled trial, which was run in the UK, and the key person, whose name you can see in the middle of the page, was a guy called Tony Hill. And he was the first person to introduce randomization to an actual medical trial, and it was 1948. He didn't do it because he had read Fisher or he had read Neyman. He didn't pay any heed to these. He just did it because it seemed the fairest way to allocate the drug or the placebo to these very ill patients. That was all. There was nothing else, nothing mystical about it. Randomization as such wasn't going to tell people what to do. And in fact, one of the key points behind Neyman and Fisher was randomized control trials, in a sense, aren't any use to you if you don't know what to do to begin with, even before you do it. Hill was just using it as a way to allocate the drug to people. And this trial showed streptomycin worked for tuberculosis. But 3 years before that, in the Mayo Clinic in the United States, people had done a control trial the way we had been doing them up till then, which was you get two groups of people who are roughly balanced for age and sex, and they gave streptomycin to to one of the groups, and they're able to show that it worked for tuberculosis. Not only that, they showed that people became tolerant to it very quickly, and some of them became deaf. They found out much more about the drug than the later randomized controlled trial did. Okay, so here's Tony Hill, and there's a quote here from him, and this is 1965, and Hill is saying, all of a sudden in recent years, everybody's been doing randomized controlled trials. After he did his first, nobody did them. During the 1950s, very few people did RCTs. It was only later in the 1960s they began to do them. And he said, look, there's one very funny thing about all this, which is that the people who are encouraging doctors to pay heed to the results of randomized controlled trials are the pharmaceutical industry. And this seemed to him to be curious. But the other thing he was saying here, which you see, is there's an enthusiasm for randomized controlled trials these days. And he's the person who did the first one, and everybody talks about him when they talk about RCTs. And he's saying they aren't the only way to work out does a drug work or not. If we ever began thinking they were the only way or the best way or whatever, we'd have gone crazy. There are other ways to look at whether drugs do work. And one of the other key things about it is he's saying it's one way to study therapeutic efficacy, which is just one thing a drug does. Recently, there was a piece of work about giving Propecia, that's finasteride, to young men whose hair is thinning. And what the study showed was that finasteride has effects on 3,500 genes. So this drug, which I think is probably typical of most drugs, they do hundreds of things. RCTs just look at one thing. So by definition, they're almost not a good way to evaluate a drug. They're a good way to evaluate one thing a drug does. Now, as I said, very few people did any RCTs at all during the 1950s. The person who is the greatest advocate for RCTs is this man you see here called Louis Lassagne. And he was the person going around the place saying that we should be doing RCTs. And he said one more critical and important thing, which was, the regulations for drugs that FDA have in the States, that's the regulator at the States, have focused on the safety of drugs. Companies have to show their drug is safe. But if a drug doesn't work, then it can't be safe. And RCTs are a way to study whether a drug works or not. So maybe the regulators, FDA and EMA, et cetera, should be asking pharmaceutical companies to prove that the drug works in addition to being safe. Now, nobody paid any heed to Lassagne about this. It took an event to change things. And the event, you probably know, was the horrific birth defects that a sleeping pill called thalidomide caused. This happened in 1959, 60, 61, and the drug was due to come on the United States market, but was held up under the 1938 Act on safety grounds. But there was clearly a crisis, and the government in the United States decided something needed to be done. And what they did was to introduce the idea to change the Food and Drugs Act so that it said companies had to prove that their drugs were both safe and effective. And in order to show that they were effective, they had to do randomized controlled trials and have positive results on 2 trials. Everybody at that point assumed that if you got a positive result on one trial, then every other randomized trial would show the same positive result. And two trials meant that for sure there was going to be a positive result. But in actual fact, we now know that maybe only half the trials you do will show a positive result. Now, one of the extraordinary things about this is that before 1962, there had only been one drug that had been through a placebo-controlled randomized trial before it came to the market. And in this trial, it had been shown to be effective and safe. There was just one drug, okay? And that drug was thalidomide. So the mechanism we put in place to stop thalidomide happening again is a mechanism through which it sailed absolutely without a problem. And the person who did that trial was Louis Lassagne, who is the person who put the mechanism we now have into the Food and Drug Act. He was the one who said there should be RCTs and there should be 2 of them, et cetera, et cetera. Okay, so there's an extraordinary irony here. Like Hill, Lassagne later changed his mind about RCTs. And here you see him nearly 20 years later. There's an article by Rossi et al. Saying what doctors think about drugs is completely irrelevant. You know, Andre Marx, who is giving a drug to a patient and says, I think this drug did whatever, you don't want to pay any heed to that. Unless you do randomized controlled trials, you really don't know what a drug is doing. And Lassagne is responding here, and he's using a tricky word for even people who speak English naturally. It's a very clever word, which is an older meaning of the word sophisticated, which used to mean to adulterate, like adding ethylene glycol into wine. That was sophisticating wines, so it's going to taste sweeter. But of course, it's not really a good thing to be doing. And he's saying that RCTs can sophisticate things, that adulterate what you understand a drug is doing. He's saying that actually, Andre, or the rest of you, when you give a drug to a patient and investigate just that patient on your own, paying heed to the patient, it's much more interesting to do and much more likely to be right. And a few years later, this is 10 years later now, you see him here, he's a good bit older, and he's saying very clearly that during the 1950s, he went around the place trying to convince people to do RCTs, but now he's going around the place trying to say to them, look, this is not the only way to the truth. And he's saying that evidence-based medicine, which has come on stream at this point, which is all based on RCTs, you know, it's really no good for a practicing doctor. We've got this person out in the waiting room. If we're trying to work out what to do to help this person, evidence-based medicine and RCTs are not going to be any use to us. So when Fisher introduced the idea of randomization, he was all about controlling the unknown unknowns, the confounders that we didn't know about. What I'm going to try and persuade you is that actually RCTs introduce confounders. If you're looking at a patient on your own, trying to work out what's going on, you're less likely to be confounded than what happens when you do an RCT. Let me show you. Right. The first antidepressant that was introduced was imipramine. This was the first of the tricyclic antidepressants. We now know that these are much more potent antidepressants than the SSRIs that came later, which are rather weak. Imipramine was introduced in 1958. And a year later, at a meeting in England, some Scandinavian psychiatrists had come over to the meeting, and they said, look, this is a very good drug. It means that we don't have to give ECT the way we were doing it before for these seriously ill patients. But, there's some people who become suicidal when they take this drug. And everybody agreed, yes, it can make some people suicidal. But melancholia has a very high rate of people going on to suicide. So even though this drug can cause people to become suicidal and even commit suicide, if you put it into a controlled trial against placebo in this group of patients who are at high risk of suicide, it would have to be a big trial. The results would turn out something like this. We would expect the drug to do better than placebo. And if you didn't know that this drug can cause people to commit suicide, you'd say, this is great evidence that this drug doesn't cause people to commit suicide, that whatever it doesn't cause people to commit suicide. Now, that's a thought experiment trial. Here's some actual trials, and these are the trials done to get the SSRIs on the market. As I said, they were weaker drugs than the older TCAs. And because of that, they had to be given to people who are mildly depressed and at very low risk of going on to commit suicide. And the results as a result of that look like this. And this is very good evidence that the SSRIs can cause people to become suicidal and actually go on to commit suicide. If you put imipramine into the same patient groups as you've got here, the results for it will look the same. And if you didn't know anything else about this drug, if you didn't know it was very good for treating people who had melancholia in a way that the SSRIs just aren't, then you would say, well, this drug causes people to commit suicide. So my point here is that RCTs tell you nothing about whether this drug or any drug can cause people to commit suicide or not. Part of the problem we've got here is you're looking at this and you're thinking these are drug trials. They're not. They're disease trials. They're condition trials. They're treatment trials. And every time a condition and a drug can cause superficially the same kind of problems, you're going to get results out of an RCT that you can't interpret. You don't really know what's going on. In the case of the glitazones, they cause heart failure. Diabetes does too. You can't tell whether the problem has been caused by the drug or the diabetes easily, at least not from the controlled trial data. In the case of the antidepressants, they can cause you to be unable to make love, or they can cause you to become suicidal, but depression can cause that as well. And it becomes terribly difficult to know what you're seeing. Is this something caused by the drug, or is this something caused by the condition? And, there's a lot of the drugs we use across medicine, almost all of them, like the bisphosphonates for osteoporosis. Osteoporosis can cause fractures. Bisphosphonate drugs can cause fractures. What's going on here? Well, actually, with the bisphosphonates, you can usually tell a bisphosphonate fracture from an osteoporosis fracture, but the same point holds. I've said these are treatment trials, and when we're trying to interpret the results of a treatment trial, it can be extraordinarily difficult. What would a drug trial look like? Well, here's some drug trials. Before the SSRIs came on the market, they were given to healthy volunteers who had no condition. They weren't being treated. So they just get the drug and you see what the drug on its own does. And we could see from these trials done during the 1980s, before the SSRIs came on the market, that they made healthy volunteers suicidal. They killed the sexual function of a lot of these volunteers. Sometimes you could be on these drugs for only 2 weeks, and potentially some of these people may have been left with enduring sexual dysfunction for the rest of their life afterwards. They also caused physical dependence. These problems vanished in the control trials, but I'm going to tell you why they vanished later. But I want to show you first, before we go on to that, before we go on to how they vanish or how companies can make them vanish, is this. Around 2004, there was great concerns about teenagers becoming suicidal on antidepressants. And the regulators, particularly the FDA in the United States, asked companies to submit their clinical trial data, not just for children, but for all age groups. And here's a page from a GlaxoSmithKline document in 2006. And this looks very bad for the company. I've taken the lower line there and just made it easier for you to see what's going on. They're saying that there's 11 suicidal acts on paroxetine compared with none on placebo. This really looks bad. And in fact, the actual data is probably worse. This is just what GlaxoSmithKline are prepared to admit to, okay? But the US cavalry is about to ride in the form of randomized controlled trials to help save GlaxoSmithKline and paroxetine, because I'm not showing you the full document here. The next line in the document shows you some data from 2 trials done on intermittent brief depressive disorder, IBDD, we'll call it. This is borderline personality disorder to lots of people, and you'll know this is a group of patients who go on to suicidal acts regularly, very frequently. And again, I've tried to make the data look a little clearer to you here. You'll see this data for paroxetine doesn't look terribly good either. But let me tell you the history here. Before this, Lilly had put Prozac into an RCT of IBDD patients and showed that it didn't work and made them worse. GlaxoSmithKline had also put paroxetine into a trial of IBDD patients, and it had tripled the suicidal act rate in this group of patients, and the trial was halted because the drug was doing so poorly. So why did they go on to do further studies, which they did? Well, the answer you're going to see on at the next page is that these further studies helped save paroxetine. We could, in fact, add 16 paroxetine suicidal acts to the 36 that you see there and still get the same magical answer, which is once you add the MDD line to the IBDD line, you get all of a sudden an astonishing result, which is paroxetine doesn't cause people to commit suicide, it stops people from committing suicide. Now that's one you have to look at and see how it's done. But really, in essence, what it comes down to is this, that depression is a heterogeneous disorder. There's a bunch of different conditions in there. And this is true for diabetes, and it's true for osteoporosis, and it's true for back pain, and it's true for Parkinson's disease and breast cancer, and pretty well every medical condition. Unless you're absolutely sure that you've got a pure group of patients, running a randomized controlled trial will mix some other patients in and potentially obscure what's going on, to the point where we can use a problem, and the company could use a problem the drug was causing to hide a problem the drug was causing. And this happens routinely in RCTs, which comes back to the point I was making earlier, that we really can't interpret what's going on in an RCT. It's much better you interviewing your patient, and that information, your sense of what's happening in this patient, is going to be more reliable than any results from an RCT. So I mentioned companies could hide problems, and this is the healthy volunteer studies of SSRIs. And you see that what it's trying to hint at here is everything's great in the Garden of Eden, but unfortunately, people aren't able to make love anymore. This is just not great. And what you got in healthy volunteer trials was over 50% of people had severe sexual dysfunction. And as I said, some may never have been able to function again in their life ever. You can get a condition called post-SSRI sexual dysfunction after an exposure of only 2 or 3 weeks to these drugs. So in the clinical trials that were done afterwards, the companies reported that only 5% of people had sexual dysfunction on our drugs, and that seems to be a risk worth taking. So how did more than 50% become less than 5%? Well, the answer is here on this next slide. which is hopefully going to play.
This is a test of selective attention. Count how many times the players wearing white pass the basketball. How many passes did you count? The correct answer is 15 passes. But did you see the gorilla? This video is from research by Daniel Simons and Christopher Chabris and is copyrighted. This is...
So, the point here is, in randomized controlled trials, we focus on a primary endpoint. You're asked, how many passes did the team in white pass the ball? All of your attention is focused in on the primary endpoint. All of the rating scales, the blood tests, everything is just focused on one thing, not on everything the drug does, just one thing the drug does. And you pay no heed to anything else. Most people assume that the primary endpoint is the commonest thing the drug does, but it's not. In the case of the SSRIs, it's not the commonest thing at all. The effect on your gut is much more common. The primary endpoint in SSRI trials is the point of commercial interest to a pharmaceutical company. And when you get investigators to focus on that, they can miss something that happens 30 minutes after you take your first pill, your first SSRI, and happens to close to 100% of people who take an SSRI. So something that happens universally and almost instantly can be missed in an RCT. So RCTs, as I've said, are not a good way to evaluate a drug. Now, I also said earlier that in the healthy volunteer trials, the companies found that people became dependent on SSRIs. And again, with a focus on a primary endpoint, this problem vanishes in the early trials. There was no mention in the label of the drugs that you could become hooked to these drugs or have problems. What do I mean by hooked? I mean, exposed to these drugs for just 2 weeks as a healthy volunteer. When you stop the drug, you can be anxious, depressed, suicidal, have nightmares, etc., afterwards. And one of the other things is that you may not be able to make love. So at the moment, partly because people get hooked to these drugs, 15% of the population in the UK and the United States, I'm not sure about Sweden, are hooked to these drugs. They're on these drugs for more than a year. When they came out at first, we didn't expect people to be treated for more than 3 months. So they're hooked to these drugs and can't get off them. And that means that 15% of the people in the UK are not making love the way they might wish to make love. In fact, if you take their partners into account, it may be 20% or more. And in some deprived areas, it could be 30% of the population who are not making love the way they might wish to. The BMJ ran an article about this 2 years ago. There was an article in the United States as well about the same thing. And they said it's down to depression. Depression is what's causing people in the UK not to be able to make love anymore. But this is not true. The depression that the 15% of people have is not severe depression. And mild depression is the kind of thing we often handle by making love, by eating more. It's not something that causes us to lose libido. And the kinds of drugs we used to use during the 1980s, the benzodiazepines, didn't interfere with our ability to make love in the way that the SSRIs and related drugs do. So we have a situation here which is actually quite extraordinary and worrying. It's a public health crisis because, you know, we are now falling beneath reproduction rates, and a great deal of that, or at least a large chunk of it is going to be down to the SSRIs, antidepressants that people are on. So RCTs greenwash medicines. In what used to be called medicine, we gave poisons to people, out of which the skill of a doctor was to bring some good. As with anesthesia, this is a poison, it can kill you, but using anesthetics with care, we can do operations so we can bring good out of the use of a poison. And this is true for all drugs. But we've got to remember we're giving a poison, and we've got to tell the patient, there's a risk you might get poisoned. So keep me alerted if anything seems to be going wrong. RCTs greenwash poisons and turn them into sacraments. What's a sacrament? A sacrament is something that can only do good. It cannot do harm. And that's what RCTs do to the drugs that we're using. And that's had a great effect on the practice of medicine, as you'll see. I'm going to disturb you a little bit further. There's a question I said I was going to ask, which is, what is data? And in one of the trials of Pfizer's antipsychotic Geodon, a man died from burns. In every controlled trial, there's about 50 articles that come out of it, ghostwritten by medical writers. None of them mention what happened to this man. The data, the figures that you look at won't show you what happened to this man. But because he ended up dead in the trial, the company has to prepare a report on what happened. And if you get your hands on that, which you can't easily, you'll see what happened was he poured petrol on himself, intending to kill himself, and set fire to himself, but died 5 days later from his burns. So his death is coded as death by burns. In a GlaxoSmithKline trial of Paxil given to teenagers who are depressed, one 15-year-old boy ended up on the street brandishing a gun, threatening to kill people, and he was picked up by the police and brought to hospital and dropped out of the trial at that point because he was brought to hospital. And again, because it was a serious event, GlaxoSmithKline should have prepared a report on that so we knew exactly what happened. They didn't. I know what happened. I can tell you that the reason he dropped out of the trial was he was caught on the street brandishing a gun and he was on Paxil. GlaxoSmithKline coded him as intercurrent illness. That is, he clearly had some other disorder going on, and this is the reason he dropped out of the trial. Well, we've got no reason to believe this unless you can interview this boy to find out, well, when they stopped your Paxil, did the problem clear up? Or interview the rest of the family, or look at his medical record to see what was his illness career like afterwards. Did things clear up, or did he have some other intercurrent illness that might explain this? You don't know what happened. And my point here is the answer to the question, what is data? In a controlled trial, people are the data. The figures, the statistical outputs, this is not data. This is other stuff. I'm not quite sure what to call it. People are the data. If you're trying to work out what's happened in a controlled trial, you've got to have access to the people and be able to interview them potentially. You might find that the person said, the data that the company calls data seemed to say the person did awfully well on the drug. But if you interview them, they say, my sex life was wiped out and I've never been able to make love since. It wasn't worth it. I would never have taken this drug if I knew then what I know now. People are the data. This is going to come back. So what randomized controlled trials do is something like this. Let's say you broke your arm, left arm, and went along to the emergency department and were met by people at the door saying, oh, good news, we're doing a randomized control trial this week of plaster of Paris. Okay, so we're going to randomly put a plaster of Paris on either your left arm, which is the broken one, or your right arm, or your right leg, or your left leg. in those circumstances, randomly applied plaster of Paris would beat placebo. But if you were to practice medicine on that basis, it would be crazy. If when people broke a limb, you randomly put a plaster of Paris somewhere on their body, figuring that this was a good evidence-based medicine practice, it would be crazy. If you practice medicine on the basis of RCTs, you're essentially letting something remove your brain and insert a program instead. And this is a problem. So part of the rest of the lecture is going to go on to answer the question, what are RCTs? Part of the problem we've got, you and I, is that in medicine as it used to be, doctors use their clinical judgment. That's changed. Your clinical judgment is now a problem for the people who run health service companies and a problem for pharmaceutical companies. And you're being told that actually your clinical judgment should be sidelined. You don't know what you're seeing or hearing or what's going on. You should be practicing according to RCTs. So that's the end of the first lecture. There's time for some questions and then a tea break or a coffee break before we go on to lecture number 2. This is our lady who's still, well, lady, I've told you, I'm going to tell you at the start of lecture number 2 that it is a lady, and we're going to go on to see what we can do to help her in the next 2 lectures.